and welcome to Teach Me in 10. Biomodal recently published a preprint on bioRxiv, and we're delighted to have time with Robert Osborne, Senior Vice President of Research and Development at Biomodal, to provide an overview of their research paper 5-methylcytosine and 5-hydroxymethylcytosine as synergistic biomarkers for early detection of colorectal cancer. So, without further ado, let's get into it. Hi, Robert, how are you doing?
Good, thank you. How are you?
Not too bad at all, thank you. So, let's get straight into it. So, what are liquid biopsy assays and where are they applied?
So, liquid biopsy assays are effectively blood tests that are just taken from a simple blood draw from typically from a patient's arm, and um, from those blood draws, you spin them down in a centrifuge. You take the plasma samples, that's the non-cellular part of the blood sample away, and then you process it through to extracting nucleic acids from it. And those nucleic acids are effectively a snapshot of what's happening in the body at any given moment in time. Because cells, as they die, are effectively shedding their DNA into the bloodstream, and then it circulates around in the bloodstream, and from there we can actually detect it and look at it.
Perfect. So, what are the limitations of current test methods?
So, one of the reasons that it's very difficult to detect cancer at early stages because there's often the tumours are very small at that stage, and therefore they're releasing much less cell-free DNA into the bloodstream. So there's a very low tumour content from amongst all the healthy cells that are dying at any particular one moment in time. In addition, there's limitations in terms of the methods that are applied, particularly the methods to identify methylation and methylation marks within the genome. And currently, most of the methods that are used actually conflate the two types of methylation marks, 5-methylcytosine and 5-hydroxymethylcytosine, into a single, what we call a single MOD-C call. And that actually effectively then masks their distinct contributions towards early detection.
Lovely. So, could you expand on the roles of 5-methylcytosine and 5-hydroxymethylcytosine as biomarkers and why they're important for that early stage detection of cancer in cell free DNA?
Sure. So 5-methylcytosine is typically found at gene promoters, where it's linked to gene repression. In contrast, 5-hydroxymethylcytosine is actually an intermediate marker in active demethylation. So that means that there's effectively a pathway from being from a region of the genome that is actively repressed by 5-methylcytosine through to being completely de-repressed and therefore transcriptionally active. So there's also differences in the distribution of these markers. So, for example, 5-hydroxymethylcytosine is enriched at active enhancers and transcribed gene bodies, and it marks dynamic epigenetic changes that occur during transitions, like transitions that occur during development and as cells differentiate. So, why are these important? Well, cell-free DNA contains methylation marks from the original cell of origin. So it's possible to, on occasion, to use those particular methylation marks and actually determine where a particular molecule has come from in terms of which cell type or which organ in the body it is derived from. Um, and we believe that being able to detect 5-methyl-C and 5-hydroxymethyl-C separately actually allows you to uncover some of the nuances around which regions of the genome are either being actively transcribed there or being repressed with 5-methyl-C.
Nice. So, you've got some powerful data that illustrates 5-methylcytosine and 5-hydroxymethylcytosine together, shows about an 85% sensitivity at 95% specificity for that early stage cancer detection. So, could you tell us a little bit more about these findings?
Sure. So we sort of entered into a discovery exercise, really, looking at the role of 5-hydroxymethylcytosine and 5-methylcytosine in colorectal cancer. So we used a product that we have that's called Duet Evoke. And Duet Evoke is a novel product that's highly differentiated within the market. It allows you to sequence not only the canonical ACGT base calls, but also separately identify 5-methylcytosine and 5-hydroxymethylcytosine. So we sequenced 69 patient samples. 32 of them were healthy. The remaining were stage one, so early stage disease colorectal cancer. And we sequenced those cell-free DNA samples from those patients using our Duet Evoke platform. Um, we then wanted to try and concentrate on specific regions of the genome that we thought might well have different signals. So we used some publicly available data, and we identified regions of the genome that are differentially methylated between healthy tissue and stage 4, so late stage colorectal cancer. And that data was derived from tissue samples. And then we identified from that approximately 12,000 regions of the genome that were differentially methylated between healthy and late stage disease. And we investigated those same regions in the sequencing data that we had derived from the cell-free DNA samples. Um, and then we built models. We use what's called lasso regularization, which effectively reduces the number of features that you need to be able to that your model has. And most of our models typically had around 50 different features in them, and a combination of about 1/3 of those were from 5-hydroxymethyl C, the other 2/3 from 5-methyl C. Um, we then kind of looked at the markers that we had, and we identified that most of them were actually in most of the 5-hydroxymethylcytosine marks were in regions of the genome that become actively demethylated. So that means that they were originally uh, hypermethylated in 5-methylcytosine at early stage, and then they progressed to being demethylated, so hypomethylated in 5-methylcytosine through the course of disease. Um, and our ability to really identify those and differentiate those using the 5-hydroxymethylcytosine and the combination of 5-methylcytosine, which allowed us to identify regions of the genome that were being actively methylated over time, led us to be able to achieve this really high level of sensitivity. So we measure these kind of things in what we call area under the curve, with the higher number being better. And we observed an area under the curve of 0.95, which is really very good for stage 1 colorectal cancer. And that outperformed any of our other models. So we also built models just based on 5-HMC only, and we got a 0.54 area under the curve, 5-methyl C only, and we got 0.7, and then the combined signal between those two, where you don't differentiate one or the other marker, which we call mod-C, we had an area under the curve of 0.66. So quite a higher area under the curve when we're able to actually separate and disambiguate these two signals.
And could you tell us why it's important to measure both 5-methylcytosine and 5-hydroxymethylcytosine in the context of liquid biopsy?
So I think really the key here is that they provide complementary insights and that they contain separate informational content that actually allows you to get an insight and a window into regions of the genome that are actively being demethylated and regions of the genome that are being actively methylated. And without that capability, it's actually very difficult to be able to achieve the kind of levels of sensitivity that we've been able to achieve with this particular study. So we built a Venn diagram in terms of the number of patients that we had that were called as having colorectal cancer at stage 1 out of the total number of patients that we actually analyzed in the study. And we compared the number of patients that we were able to correctly identify using the combined 5MC and 5HMC model, and then compare it to the number of patients that were correctly identified using a 5MC model only or a 5HMC model only. And what we found was that we were able to identify 15 patients that had detection in 5MC and 5HMC model and the 5MC model, but not the 5HMC model. So those were 15 patients that one could detect using a signal alone from 5MC, but would be missed if you looked at 5-HMC on its own. And then we did the reverse as well. So we looked at patients that would be correctly classified if we looked at 5-HMC models and compared that to the number that we observed with a 5MC and 5-HMC model, and then the number that we would detect if we looked at a 5MC model only. And there we found 13 patients that were detected using the 5-HMC only model and the combined model as well. So these kind of 15 and 13 patients that were found either on 5MC or 5HMC sort of indicate that really the informational content in these two marks can be separate and can be used to identify different patients. And this is obviously contributing to a high level of sensitivity.
Incredible. Well, thank you ever so much for talking to us today at Teach Me in 10.
Thank you very much.
Thanks so much for watching this Teach Me in 10, and I hope I'll see you again very soon.